question is, a fly with an apricot colored eye was crossed with sepia-eyed fly of opposite sex. Let's say the female and the male. In the F1, all the flies were wild type. The genes responsible for the two phenotypes were, it is saying, in the F1 generation, when we are crossing these, in the F1 generation, all of them were of the wild type, right? So it is asking us the genes responsible for the two phenotypes are. Try out the question. Two different type of phenotypes. First one is allelic, second non-allelic, third pseudo-allelic, fourth paralogous genes. Try out the question. Easy one. 1 minute for this. Got colored eye, and the other one is having sepia colored eye. In the F1, we had all of the progenies formed being of like complete. We have two different phenotypes. F1 was wild type, so it is asking us what is the reason for having two different phenotype, because they are non-allelic in nature. Different type of genes are involved, that's why we are having non-allelic nature. This cannot be allelic, this cannot be pseudo-allelic, or this cannot be paralogous genes. It is asking us about why it is having two different phenotypes. Reason being, the genes are non-allelic in nature. That is, two different genes are involved, which is causing this particular characteristic. B is the correct answer. Not a difficult, like it's a very simple question. Next question, give it a try. Again, very easy question. Understand the question, and if you were there in the developmental biology class, we learned about the exact similar thing. So try out the question, everyone. Look into the question. In a transplantation experiment, area of the presumptive ectoderm from an early frog blastula was transplanted from to a region of a nuid grastula. Okay, from a frog to a nuid, destined to become the parts of the mouth. The resulting salamander larvae had frog-like mouth parts instead of the balancers that are observed during the wild type of your nuet embryo. You have a complete different organisms altogether. This is an example of what? This is an example of it cannot be determination, regional or autonomous. Over here, we are changing what? We are changing the genetic makeup, we are changing the complete genetic thing, right? And because in the even after moving to some other area in the new organism, it is still forming frog like mouth parts. That is, it is still forming the same genetic content which it had to form in the previous one. So it is a type of your genetic specificity of interaction. Those of you who were there in the class when we discussed about all these things, we learned about this genetic specificity also. There was one small information which I shared about it, and the question is from that similar concept. Remember, B is the correct answer. See, in this paper they have given this question in part B, but this question holds very good potential to be asked in part C as well, right? Because it's a very interesting question given to you. It's just a matter of you reading the question properly and understanding the question, that what is it actually asking you. Okay, B is the correct answer. Give yourself 2 and 4 markers accordingly. Next question. In which of the following stages of Arabidopsis embryogenesis do the visible distinction between the adaxial and the abaxial tissues of the cotyledons become initially apparent? That is, you can easily see a clear distinction between the abaxial and the adaxial tissues. Which stage is it? Is it globular, zygotic, torpedo, or the mature one? Try it out. At which you can easily distinguish between the two things, right? You can easily distinguish between the adaxial and the abaxial tissue. That is the tissue that are present on the lower surface and on the upper surface. Remember about this. C is the correct answer. Next